Welcome to our YouTube class on glycolysis. Glycolysis is a central metabolic pathway that occurs in the cytoplasm of cells, where one molecule of glucose is broken down into two molecules of pyruvate, generating energy in the form of ATP and reducing equivalents in the form of NADH. It is a crucial process in both aerobic and anaerobic organisms, providing energy for cellular functions and serving as a precursor for other metabolic pathways like the citric acid cycle and fermentation. Phases of glycolysis. Energy investment phase or preparatory phase. This phase consumes ATP to activate glucose, converting it into a form that can be readily broken down. Energy payoff phase. This phase generates ATP and NADH, producing pyruvate as the final product. Steps of glycolysis. Energy investment phase. Step 1. Hexokinase reaction. Here glucose is converted to glucose 6-phosphate. Hexokinase is the enzyme helping for this. Here ATP is also converted to ADP. This phosphorylation of glucose traps it inside the cell, since glucose 6-phosphate cannot easily cross the cell membrane. Step 2. Phosphoglucose isomerase reaction. Here, glucose 6-phosphate is converted to fructose 6-phosphate. Phosphoglucose isomerase is the enzyme helping for this. This conversion of glucose to fructose is to prepare for further phosphorylation. Step 3. Phosphofructokinase 1 reaction. Here, fructose 6-phosphate is converted to fructose 1,6-bisphosphate. Phosphofructokinase 1 is the enzyme helping for this. Here, ATP is also converted to ADP. This is the rate-limiting and most regulated step of glycolysis. PFK1 adds a second phosphate group to fructose, making it more reactive. Step 4. Aldolase reaction. Here, fructose 1,6-bisphosphate is converted to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Aldolase is the enzyme helping for this. Here, cleavage of fructose 1,6-bisphosphate occurred that is into two 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Step 5. Triose phosphate isomerase. Reaction. Here, dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate. Triose phosphate isomerase is the enzyme helping for this. Due to the conversion of DHAP to G3P, there are two molecules of G3P available to continue in the pathway. Energy payoff phase. Step 6. Glyceraldehyde 3-phosphate dehydrogenase reaction. Here, G3P, NAD+, and phosphate is converted to 1,3-bisphosphoglycerate, NADH, and H+. Glyceraldehyde 3-phosphate dehydrogenase is the enzyme helping for this. This oxidation step generates NADH and attaches a high-energy phosphate group to the G3P. Step 7. Phosphoglycerate kinase reaction. Here 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate. Phosphoglycerate kinase is the enzyme helping for this. Here ADP is also converted to ATP. This is the first ATP generation step, where a phosphate group is transferred to ADP to form ATP. This is an example of substrate-level phosphorylation. Step 8. Phosphoglycerate mutase reaction. Here 3-phosphoglycerate is converted to 2-phosphoglycerate. Phosphoglycerate mutase is the enzyme helping for this. This is the rearrangement of the phosphate group from carbon 3 to carbon 2, preparing the molecule for dehydration. Step 9. Enolase reaction. Here 2-phosphoglycerate is converted to phosphenylpyruvate and a water molecule. Enolase is the enzyme helping for this. This is the dehydration step that forms phosphenylpyruvate, a high-energy intermediate. Step 10. Pyruvate kinase reaction. Here, PEP is converted to pyruvate. Pyruvate kinase is the enzyme helping for this. Here, ADP is also converted to ATP. This is the final step, generating another ATP molecule and producing pyruvate. In this pathway, 2 ATP are consumed in steps 1 and 3. 4 ATP are produced, that is 2 ATP from each G3P, in steps 7 and 10. The net ATP yield is 2 ATP per glucose molecule and NADH yield is 2 NADH per glucose from step 6. Regulation of glycolysis. It is regulated to ensure that energy production matches the cell's needs. The main points of regulation are hexokinase, inhibited by its product, glucose 6-phosphate. Phosphofructokinase 1, allosterically regulated by ATP and AMP. It is the most important control point of glycolysis. Pyruvate kinase, inhibited by ATP and activated by fructose 1,6-bisphosphate. Fate of pyruvate. In aerobic conditions, pyruvate enters the mitochondria, where it is converted to acetyl-CoA and enters the citric acid cycle. In anaerobic conditions, pyruvate is reduced to lactate in animals or ethanol in yeast via fermentation, regenerating NAD+ for glycolysis to continue. Importance of glycolysis. Provides a rapid source of energy, especially in anaerobic conditions. Supplies intermediates for other biosynthetic pathways, such as amino acids and nucleotide synthesis. Occurs in almost all living cells, highlighting its fundamental role in metabolism. Thank you for watching. Please like, share, and subscribe to our channel for more informative videos.